Rhomboid proteins comprise a superfamily of serine protease enzymes with active sites within the cell membrane. They are present in almost all organisms and play a critical role in cell signaling by freeing signaling proteins, cleaving precursor membrane proteins into their active components and releasing them from the membrane. These cleaved signaling proteins, such as epidermal growth factor, regulate cell events, including developmental signaling, wound healing, and, critically, the epithelial mesenchymal transition that occurs in many cancers. Our presentation will discuss a recent study about the unusual diffusion of rhomboid proteases within the membrane and the implications for the structure and function of this protein family. But why is internal protein diffusion even important? Protein diffusion within the membrane is critical in order for the cell to respond to stimuli and replenish damaged areas. Diffusion allows for a faster response to changing conditions by increasing the likelihood of a protein-ligand interaction. A standard model of diffusion within the membrane is the Safman-Delbruck model, which shows that larger, multi-spanning integral membrane proteins diffuse at a slower rate than smaller, single-spanning membrane proteins. To study how rhomboids move within the cell membrane, the Urban Lab at Johns Hopkins University tagged membrane rhomboids with fluorophores and used single-molecule turf microscopy to track how these proteins diffuse in various cell lines. Each of the rhomboid proteins examined diffused faster than any previously measured multi-spanning membrane protein, including the iconic retinal pigment rhodopsin. Researchers found that diffusion rates varied for different rhomboids and different local environments, but all of them diffused exceptionally quickly. By making a synthetic membrane and putting single proteins inside, the researchers could fine-tune experimental conditions. Two variants of GLPG, the best classified rhomboid protein, diffused faster than the theoretical approximation defined by the Safman-Delbruck limit, suggesting that rhomboids do not adhere to typical membrane dynamics. Crystal structures show that GLPG has a thinner hydrophobic core than other membrane proteins, so the hypothesis was that GLPG structure distorts the lipid bilayer to promote faster diffusion. Integral membrane protein diffusion is generally slower in thicker membranes and faster in thinner membranes, but GLPG demonstrated the opposite effect relative to control proteins. By engineering a mutant GLPG that lacked the cytosolic N-terminal domain, the researchers hypothesized that the proposed distortion effect on the bilayer would be attenuated, slowing rhomboid protein diffusion within the membrane. When tested, they found that diffusion rates were significantly altered, indicating the proposed lipid bilayer distortion effect is real. To test whether diffusion was the rate-limiting process in any rhomboid-mediated pathways in the cell, the hydrolysis rate of signaling proteins was observed after adding a drug that doubled intramembrane protein diffusion. Proteolysis rates increased threefold, suggesting that diffusion regulated reaction rate. Likewise, slowing the process down by attaching beads decreased the rate significantly. Through evolution, rhomboids have been tuned to overcome the limiting conditions of their environment. Because rhomboids only produce a single signal molecule each time they encounter a target protein, they need to find as many target proteins as possible in the shortest amount of time in order for signaling to be effective. In contrast, other classes of serine proteases do not have to overcome membrane viscosity before catalyzing reactions, and are limited by their own catalytic efficiency instead. As such, this proposed model of diffusion presents a new way of examining protein membrane dynamics, especially for other integral membrane proteins. Alongside this, advancements such as cryo-EM have made visualizing the structures of proteins easier, and several rhomboid proteases have a well-determined crystal structure. This paper critically ties together the irregular structure of the rhomboids with their diffusive functionality. Hence, we are presented with a structural characteristic that helps find other proteins that will diffuse similarly in the membrane. Notably, rhomboid proteases are implicated in many clinical pathologies such as cancer, Parkinson's disease, malarial infection, and type 2 diabetes. Understanding the complexities of rhomboid proteases and the conformational changes they enact is crucial to the creation of new and effective therapies that can specifically target these proteins.